Okay, kia ora. Welcome everyone. It's great to have a great turnout. So I'm Liz Ledgewood and I'm here as Deputy Head of Department, standing in for Peter Dearden, who apologises because he's in Japan at the moment. But it's my really great pleasure to introduce our speaker today for what his talk on his PowerPoint, the file name's, I think, final presentation. Is that right? So I think many of you, I mean, we can say Warren needs no introduction, which is partly true, but I think for many of you, you probably don't know much about Warren's history. I'm not going to spend much time talking about it because, one, we have a celebration in a couple of weeks coming up where we'll have more to say, but also because Warren says he's got less than a minute per year. So we want him to get started. So Warren joined the department in 1968 as a PhD student. And I actually did go to find his thesis, but unfortunately, when we read it upstairs, they're all jumbled in a cupboard. So I didn't manage.
I've got it in my office.
You've got it in your office. Yeah, you've got a copy. I should have come to you, shouldn't I? So his, at least in his record, it says his PhD was studies of nucleotide sequence arrangement in bacteriophage DNA. And so I think that tells you that Warren has been really here since the beginning of really molecular biology, and certainly in this department, and has been a crucial part of it. Warren has won many awards and medals over his career. He was actually made, I guess he spent almost his entire scientific career in this department, apart from 3 years as a postdoc at the Baylor College of Medicine. He was appointed as a lecturer in 1975. He was then appointed professor actually in my honours year, so I do remember this, in 1989. And at that time in particular, he was, I think, one of the youngest staff members to ever be appointed professor at this university. So that was quite an achievement, and I do remember the excitement of that when I was a student even. Since then,
like yesterday.
Does seem like yesterday, I agree, but it's not. I still feel young because Warren obviously lectured me and many, many people in this room, but I think that's not the case either. So Warren has made a huge contribution to the biochemistry department. He's mentored and supervised and helped many, many students. Many have gone on to become academic staff here and in many places around the world. And so I'm going to leave it at that for now. His CV now says his research specialisation is neurological disease, chronic fatigue syndrome and long COVID. But actually, when I think about Warren, I think about the ribosome. And I think today you're going to take us through that career. So I'm going to hand it over to you, Warren. We're really looking forward to hearing you speak.
Thank you very much. So I came into this building the first day it was commissioned, and I was the first PhD student in biochemistry to have my role in the building. So I think what I'd like to do is dedicate this lecture to all of the people that have been here in the building over my time, and particularly colleagues who created a wonderful environment for us to do creative work here, and especially all of the students who were postgraduate students that have contributed to the work that I want to talk about. So I've seen this lecture on the Central Dogma, Crick's original Central Dogma that was published in 1958. I first learned about this as an undergraduate student at Victoria University, very briefly, and that there was a new field of molecular biology starting. But actually, this really underpins all of the two main research themes that I've been working on. Firstly, the relationships between DNA and RNA and protein and information flow that leads from the DNA to the synthesis of a protein. That's what I'm really going to focus on today. But also in my work on understanding the pathophysiology of unexplained human diseases, where I've used large DNA data sets and RNA data sets and protein data sets, so applying the Central Dogma to understand that. So it's sort of dogged me through my whole career. So what made me think about coming to Otago? So I really want to use a kind of modern idiom and say there were two major influences. One was Merv Smith. Now, I heard about Merv Smith when I was doing honours at Victoria University. a student from Otago who was in his lectures said, we've got this wonderful, enthusiastic, inspiring molecular biologist. And lo and behold, when it came to the examiner coming up for our honours program, it was Merv Smith. And he did a rather strange thing. He went through his process and then sat down with each individual student, much to the horror of the academics at Victoria, and critiqued them face to face on both their papers and their thesis. Now, for my thesis, I had purified a protein urate oxidase, the one we're missing, so it makes us susceptible to getting gout. And I found that there was copper involved in the enzyme, and the crowning glory was the last experiment I did out at the DSIR in Gracefield Lower Hutt, which had electron spin resonance. And I was able to show that the copper atom was switching from a copper state 1 to 2 during the reaction mechanism. So I thought I'd done a pretty good thesis. When Merv sat down with me to talk about my thesis, he brought me down to earth. He said there was a major deficit in your thesis. You hadn't done a sucrose gradient. I had no idea what a sucrose gradient was, and I didn't dare tell him that. But what I found out subsequently was Otago biochemistry had taken had got an ultracentrifuge, and he was working on DNA complexes from a bacterial virus and isolating his DNA complexes by sucrose gradient. And what I learned subsequently about Merv is when you talked about academic or progress with him, he always made a very challenging statement first. And it was really aimed at wanting you to think even higher, even if you were doing quite well. And so I valued that through all my career. But anyway, I was stung by that comment. And so I came down in the summer of 1967 and to do some sucrose gradient experiments, got some data on my protein and went back. So I'd sort of seen what the department was like. The other person that was a major influence, of course, was George Peterson. And I had a person who was always next door to me in undergraduate labs at Victoria University because of our surnames. And she had gone to Massey to do a master's, and George Peterson was her secondary supervisor. And she said to me, she's now my sister-in-law, she said to me, don't decide on where you're going to do a PhD until you've talked to George Peterson. So I went up to Palmerston North, where he was, to talk with him. And George, as I understand, did one of his characteristic things too. He profusely apologized to me before we even started the conversation, and then said, oh, look, I'm sorry, I can't actually be a supervisor to you at Massey. I'd never even mentioned Massey, because I'm going to Otago next year to be head of Department of Biochemistry. I thought, wow, the stars are all aligning. What's more, the crowning glory was the student that had told me about Merv I'd had a romantic interest in. And so that actually just tipped the balance. So Merv Smith and George Peterson then were the head of departments here for the first 30 years of my time here. And so they've been an enormous sort of influence. Now, so I came down then to be the first PhD student of George Peterson when he took over the HOD-ship at the beginning of 1968. And doing a PhD then was quite different from now. We had no computers, of course. There were no major pieces of technology. There were no large data sets. What a student had to do was to get into the research lab and spend hours in the research lab trying to do good experiments. And the only time you sat down at your desk was briefly to plan the experiment. And when you were exhausted at the end of the day, hopefully having accumulated some data to do something which was a moment of joy, where you took out a piece of graph paper and you could plot the data points that you had achieved during the day on a graph. And I've taken this out of my PhD thesis here just to illustrate kind of the beauty of some of the data that we derived. So I was using very primitive DNA sequencing technology. It was before Sanger sequencing, degrading DNA with diphenylamine and acid and releasing pyrimidine tracks. And so each of these 3 profiles on the left are the 3 bacteriophage DNAs that I was working on. And so a tract would come out according to the number of pyrimidines in it, and then you'd wait about another 6 hours and another one would come out. And these were all radioactively labeled, so I'd get all of the data. So this was a 6-day elution to get one of these profiles. And then you could take each of those peaks, and I've highlighted there the one that has 8 pyrimidines, and run it on another column and separate the cytosine and thymine components. And so from those 3 profiles on the left, there were 40 columns to be run to generate all of the other data. So, you know, that was kind of quite intense laboratory work. However, this was a good, successful PhD. I got a number of publications out. And just amusingly, for those that know George a little bit, that when I left on my scientific OE, for the last 2 papers, I drafted the paper, and in those days, the diagrams were drawn in Indian ink on cardboard. And so I left with George to critique the manuscript, and he said he would submit the paper. So 6 months into my OE, scientific OE, I'd heard nothing. And so I thought I need to write to George and just remind him that he was going to submit the paper. So how would I do that graciously? So I said, I wrote a letter and said, I know how busy you are and whatever. You know, if you're too busy to submit them, why don't you send me the diagrams and I'll submit? I didn't expect him to do that. It was just hopefully jog him along. And about 3 weeks later, the secretary where I was came in and she said, there's a huge package has arrived to you from New Zealand. It was about that big by that big. And George had sent me the diagrams. But there was a happy ending to it because I submitted the papers, and maybe it was because I was at a fairly prestigious place in the United States. Both papers went in without any revision. So at the end of my PhD, one of the things I was thinking about, what would I do to continue on in my career? And I thought that, you know, New Zealand had 2 prominent people working on DNA. George, of course, and the other person was a molecular biologist at Auckland University called Peter Bergquist. He was very self-focused and very strongly opinionated. And I thought, there's no way I'll be able to establish an independent research career back in New Zealand if I worked on DNA. You see how much foresight I had in those days. So, but I was inspired by a paper in Nature by a young postdoc who had come from Yale to Cambridge, Joan Stites. And what she had done is discover the signal in messenger RNA for the start of protein synthesis. It turns out to be AUG, which is the codon in the genetic code for methionine. And it just struck me, protein synthesis was a newly kind of breaking area. This would be a good thing to go into. Now, I got a photograph of her from the internet today. You can see that today, aged 83, and you can see she still has the same kind of energy look in her face as she did originally. But what I particularly noticed, she still had the same hairstyle. On the right there, I've got a picture of her husband, Tom Stites. He was in the same class as her at Yale, and he was originally an X-ray crystallographer working on polymerases, but sometime in the 1990s, he switched to the ribosome and got the first structure of the 50S subunit of the ribosome just before the international meeting in Queenstown that I was hosting of the ribosome, the first in the Southern Hemisphere. So this was, the right-hand picture there was a picture taken by an Otago Daily Times photographer when we were being interviewed beside the lake in Queenstown about the conference. He subsequently won the Nobel Prize in 2009 as one of the 3 people who got it for resolving the ribosome. So I'm kind of happy to give that picture to my grandchild as I'm standing beside a Nobel Prize winner. Okay. So what I did then was to look at protein synthesis areas and signal, messenger signal areas. And I didn't want to work in an area where there were too many groups in the United States because then I couldn't establish my own niche if I was working back here. And there were lots of people flocking into the initiation phase of protein synthesis, quite a few in elongation, and just two really good groups working on termination, the last stage. So I thought maybe that's where I should go and I can establish a niche. So I wrote to one of these was a young clinician, Tom Kasky, who was working at NIH in Marshall Nierenberg, the Nobel Prize winner for the genetic code in 1968 at NIH. So I thought this would be pretty cool, going to NIH, working in a Nobel Prize winner's lab and with a young clinician. Unfortunately, it was a deja vu from seeing George, really. A couple of weeks before I was due to leave, I got a letter from him saying, I've just been asked to establish an Institute of Molecular and Human Genetics at the Baylor College of Medicine in Houston, in the Texas Medical Center, and I'm moving there. And you're very happy to move with me there. I hope you will, but I'll understand. And so I had to it was a bit disappointing that I was missing out at NIH and with Marshall Nierenberg lab, but I decided to go there, and it was a good decision. Life was quite different, how from doing a PhD at Otago and there, and I realized this from the first day. The young student that had told me about Merv Smith, who was now my wife, and I arrived in Houston, and we stayed with Tom Kasky, and I was kind of rudely awakened the next morning at 5:30 in the morning by Tom to say, we're going into the lab just after 6. And I realized that actually the way that his group worked is he would try and always get into the lab by 7 a.m. His group would scramble in a minute or two before, half of them still in their pajamas. And then at the end of the day, he would tend to go about 7 p.m. and people would all be nervously waiting for him to go. And then there would be a mass exodus. So for the first few months, I really felt, I was under trial, but fortunately I managed to get a first author PNAS paper in the first 10 months. And then after that, everything relaxed, and I really developed a very good relationship and got a number of very good papers out of that experience. So in the last part of my time there, I was thinking about, well, what could I take from this to establish an independent research career? And what I noticed was in the United States, all of the people working on protein synthesis were just working on the solution chemistry. They weren't working on the biology at all. They just threw a bit of slimy ribosome into their test tubes, not knowing what it was really doing. So I thought this was a bridge that I could actually cover by working on sort of both areas. And I discovered that there was a Max Planck Institute in Berlin that was actually taking the bacterial ribosome apart and finding what all the parts were and raising antibodies to this. So for the last, in the last year, I spent 7 weeks in Berlin, just immersing myself in the ribosome there. I remember Tom's last word. It was a bit like Merv's words. He said, don't come back until you've got some data. So this Institute for Molecular Genetics was headed by an outstanding scientist, Heinz Günther Wittmann, and I discovered when I got there that everybody were terrified of him. So I learned something then, that if you play the dumb, naive Kiwi in situations like this, it gets you a long way. So I went and saw Wittmann and said, look, told him why I'd come, and asked him if I could come and see him, maybe once a week and just talk about what I'd done and get his advice. And he really warmed to that. And so I had developed a close relationship with him right up to the time, unfortunately, he died in 1990 of lymphoma, and that helped my career development. The one interesting thing there is because they had isolated all of the individual proteins of the ribosome and they were developing antibodies, they had about 200 New Zealand white rabbits, so I felt at home, and a flock of sheep actually in a building associated with the institute. I didn't feel quite as home with that. But that's how they got their antibodies. So I did some work with the antibodies. So from that, I came back then to Dunedin with a number of questions that I wanted to study as an evolving independent research career. The first was that I'd got interested in the ribosome catalytic center from my exposure in Berlin. And so part of the termination event is to release the completed polypeptide, and that's done by the catalytic function of the ribosome, just like peptide bond formation, which is the main part where the catalytic center is. So I wanted to try and work out what the catalytic center was all about. Secondly, of course, bridging the gap between the chemistry and the biology, what parts of the ribosome mediated the mechanism of termination of endoprotein synthesis in both bacteria and mammals. And then, because Tom Kasky claim to fame originally, he'd shown that proteins rather than tRNA decoded the stop codons of the genetic code, wanted to know whether the decoding protein acted like a mimic of a tRNA or something else. And so that was a third question. So I want to just sort of address those now. So I first made contact again with a scientist at the Max Planck Institute who had just managed to reassemble the large subunit of the ribosome from all of the individual ribosomal proteins and RNA that they had isolated at the Institute by just doing a bit of heating and adding a bit of salt and another heat step and then some magnesium. And this here you'll see in the middle at the bottom is a primitive electron micrograph, and the right-hand part of it is the reassembled ribosomal subunit. So what I wanted to do, I saw would be very useful to try and pin down the catalytic enzyme of the ribosome. People told me you'd get a Nobel Prize for that. Ironically, you'll see later in the talk that did occur, but not the way that I'd planned. So I wanted to prepare, they knew thought then there were about 34 ribosomal proteins. So I wanted to prepare 34 individual ribosomes, leaving out one protein at a time, and then to test each of these and for their activity so that I could narrow down at least what might be the catalytic enzyme. Hit a bit of a stumbling block when I actually went into Knut Niehaus's lab, because I found that I had to undergo a severe test before I was able to start the collaboration. That is, I had to work with his expert reconstitutor of the assembly of the subunit, and had to get an activity of the ribosome close to him. Otherwise, no, I wasn't allowed to use all of the materials. So we worked for 3 days, firstly isolating the RNA, then doing the assembly, and then testing the functions. Fortunately, this guy, Hartmut Schutzer, was much more chilled than his boss, and he knew what was going on, and so he slacked around a bit. And so my ribosome was 50% more active than his. So I got my ticket to go. So when I did this then, we actually managed to find 4 ribosomes that were lacking a protein, which had lost the catalytic activity, both catalytic activities for termination and peptide bond formation. And that's L2, L3, L4, 16, L for large subunit, 4 proteins. What is more, there were other of the ribosomes I'd prepared lacking a protein that had interesting characteristics as well. I won't talk about those today, but which I followed up with. So then a PhD student, Vicky Sumpter, started with me, and her role was to find the catalytic enzyme. And the model that was available from the Niehaus lab was that one of the ribosomal proteins was likely to have a histidine that was involved in the catalysis. So there'd be nucleophilic attack, electron withdrawal. You'd break the bond between the tRNA and the growing peptide, and that growing peptide then is linked in a bond to the new amino acids which come on. That's peptide bond formation. The hydrolysis event of termination, instead of see if I can find this, where are we? Instead of the new amino acid here, you just have water. So the growing peptide or completed peptide then is just added onto water, and then you've got your completed protein. So what was known then, enzymes which use this kind of mechanism could be inhibited with the RNase inhibitor diethyl pyrocarbonate, and that would attach to that nitrogen shown by the arrow. And so Debbie modified these proteins that we'd identified as being important, and with the unmodified, reconstituted the protein in the ribosome that was missing that protein and tested their activity. Now, I had some evidence that L16 was probably the most likely, so she tried that first, and the modified L16 had no activity. We were ecstatic and very tempted to say, we don't want to do the other ones because we found the catalytic enzyme. I knew, of course, I couldn't do that. So unfortunately, the ecstasy just diminished as she did the other three, because when she did it with L2, she got the same result. Modified L2 lost activity, modified L3 lost activity, modified L4 lost activity. So I'd planned a Nature paper from this work, but with heavy heart, I wrote a paper up and sent it to a European journal. And again, a little bit of light at the end of the tunnel, because one reviewer said that no other reviewer ever said in his opening sentence, this paper must be published. I think he felt my angst, he or she felt my angst in writing the paper. And so we got it published. So I want to just fast forward now 20 years when the first ribosome structure came out. So this was an image in the, I think, supplementary material from a Venki Ramakrishnan paper from Cambridge. And what he's demonstrating here is the protein and the RNA distribution. And so this is the inner face of the large subunit where the catalytic center is. And what you can see is the proteins, the black blobs, are all stuck around the outside. Interestingly, on the back side, it looks like someone who's good at design has put the proteins on because they're equally spaced on the back side, you can't see. But here they're around the outside. And the gray areas is ribosomal RNA. And we knew by then the catalytic center was just under that central protuberance. And as you can see, it's gray, it's not black. And so that said that the catalyst of the ribosome was not a protein, but was intrinsic to the RNA. In other words, it was a rare RNA enzyme. So of course that explains why we weren't able to find the catalyst. I must say, I went back to see if I could find the original paper I'd published, and I found the original manuscript was in the back of one of my filing cabinets, got it out, and in the discussion I could see the tears on the discussion page. And when I read it, I have to say I felt quite proud, because actually what I'd written there was quite insightful for the original thing. So since we're 20 years forward, I just want to take an aside with a student that came aboard. Harold Bernhardt was a PhD student, was a honours student in the department in the 1970s, and went for many years to be the department manager for anatomy. And he came and saw me one day and said, you know, would I take him on to do a PhD on the origin of life? He was obsessed with the origin of life, fascinated perhaps I should say, but really obsessed. So I don't quite realize how the context, I can't remember the context, but I was trying to persuade him the best thing for him to do would be to do an RNA study, because he was interested in the RNA world, on a modern sort of thing, so that there would be a career development for him, because I didn't see that if it was done from a point of origin of life, that restricted a lot his chances to go ahead. So we travelled back from Queenstown to Dunedin, arguing this, very animated, and somehow I found myself in Wanaka. We were arguing so much that I'd missed the turn-off. But by the time, fortunately with the extra distance, by the time we'd got back, I'd persuaded him to do this. And then at the 11th hour, a day before he was going to start his PhD, came to see me. Would you still supervise me? I did an origin of life. So I caved. And it turns out, actually, that took me into areas of ribosomology, the origin of an RNA ribosome, the dawn of coding. We published a number of theoretical papers here together, which were really quite exciting. I insisted he do some experimental work. They aren't shown on the screen there because they weren't quite so exciting, but they were important for his PhD. So that was an interesting sort of, these things come along and you've got to sometimes make the right decision. And I made the right decision to accept him. So one of the questions I asked was, does the decoding release factor, a protein recognizing stop codons, mimic a tRNA? And there was always a possibility that the release factor, this protein, might just bind to the ribosome, cause a conformational change, and the decoding was still an RNA decoding between ribosomal RNA and the codon and the message. So in one of my trips to Berlin, I made an RNA with T7 polymerase and substituted uracil for 4-thiouracil. So the stop codon was thio-UGA, and I radio-labeled some release factor with 35S-methionine and assembled a protein complex, and ironically needed to do sucrose gradients to actually work out what was going on. But I was able to show that activating the zero-link cross-linking from the 4-thio-U, it cross-linked to the factor. So we knew that the factor was directly contacting the factor. So then John Moffat came on board with his PhD, and he was looking at trying to determine the domains of the factor, and we concluded there were at least two domains. One seemed to be important for catalysis and one seemed to be important for codon recognition. And there were small peptide motifs, the GGQ, for example, seemed to be important for hydrolysis. SPF was associated with decoding. So what we're looking at there in this model is that with the two tRNAs, it's in the space between the two subunits spanning the catalytic center at the top of the large subunit to the decoding site at the bottom of the small subunit. And we're hypothesizing that the release factor, the protein release factor, would be a highly elongated protein, which would functionally and structurally look like and be doing what a tRNA is. That was the hypothesis. So how to test this? Well, there was a very bright scientist from Santa Cruz in California, who I think should have got the Nobel Prize because he did a number of important things in ribosomology. And he had developed this technique called hydroxyl radical mapping. In fact, it's the only lab that had used it and has used it apart from ours subsequently. And what this was that you could attach this reagent, which we called BABE, onto cysteine residues, and he was using it to map where proteins were in relation to the RNA that they were sort of bound to, because remember, we didn't have any structures. This is back now at earlier times. And so with iron caged in the reagent, we could activate the Fenton reaction with hydrogen peroxide and it would produce hydroxyl radicals. So this was like a missile launcher. It launched the missiles, which are the hydroxyl radicals, and they broke the RNA that was close to the site where they were launched. So this seemed to be a perfect method for us to test our hypothesis. So I had a research fellow working for me, Kim McCorn, who had done a PhD in organic chemistry. So she disappeared off to the chemistry department for 9 months and came back with a babe. And so we were able to use that reagent to test our model. And Debbie Scarlett did her PhD on this. And what she first had to do was to take cysteines out of the release factor and replaced them with neutral alanines. And we held our breath. In fact, there were 2 cysteines in it. Held our breath that it would retain function. And what that image is supposed to show is that a very excited PhD student, because it remained functional. And then she introduced individual cysteines near the SPF and GGQ motifs, several in individual modified factors. Attached babe, so we attached our missile sites there and then fired off the missile. So those 4 steps took her about 2 years, and I thought, wow, this is going to be the shortest PhD I've ever supervised. But the last step took another 2 years to actually work out being able to find out where the cleavage sites. It was really tricky, and I understand now why no other lab other than the NOLA lab has actually used that. So what she had to do was isolate the RNA, use reverse transcriptase to get a DNA copy, do Sanger sequencing, but we could only get good output signals if we used a certain ratio of 32p and 33p in the Sanger sequencing. And then you could see darker bands where the cleavage had occurred. But the data that came out was absolutely clear cut. With the cysteines that she'd put on the near the SPF, the cleavages in the ribosomal RNA, and that's the left image there. You can see hopefully red and blue and magenta, and these were cleavage sites from a couple of different cysteines that were individually attached. And only cleavage sites there, and that happens to be the decoding site just below the head of the small subunit. There was no cleavages in the large subunit RNA, but when she had the cysteines put on the GGQ, near the GGQ, all of the cleavages were around where we knew the catalytic center was. So this said that the GGQ was near the catalytic center and the SPF was near the decoding site. And this seemed to confirm our model, our proposal. Now, this was at the end of 2001, and I was hosting the International Ribosome meeting in February 2002. So I was very happy that we'd got some data, really good data to take to the conference. You should never get too confident, because a structure came out in December of 2001 of the release factor. And for us, it was really ugly structure. It was compact. And the distance between the GGQ and the SPF was only 22 angstroms, not 76 as our model proposed. So somehow I couldn't think how our derived data was incorrect. So I was hoping that she had trapped in the crystal structure a more stable form of the factor, which was not its functional form. So she was coming to the conference, presenting a paper, but her abstract said nothing about this. So I was pretty nervous when she gave her talk, and she did talk about the release factor, but a cryo-electron microscopist who was developing cryo-electron microscopy on the ribosome and looking at each of the protein synthesis states, trying to build up a video of the states of protein synthesis. This was Joachim Frank. He became known as the father of cryo-electron microscopy and got the Nobel Prize for us in 2017. He got up at the end of her talk and said, I've just done a cryo-EM study of the release factor on the ribosome, and it doesn't look anything like your structure. I can't remember whether I actually leapt up out of my chair, hailed and put my hand out, or whether it was just in my mind, but that was the effect. So the publication of his model came out a couple of years later, and you can see here, here's the GGQ, here's the SPF, and that's 76 angstroms apart. In fact, it told us what was going on, because if you took the compact solution structure and you unfolded this, where are we? This domain here, like the derrick of a crane, you got this structure. And I think you can see that rather looks like the structure that had been, the image that had been evolved from the cryo-EM structure. So that was good. I put on this slide another interpretation of some X-ray crystallography from Venki Ramakrishnan's group, which was just magic to me. I got a paper to review of his. This was in the supplementary paper. For me, it was the most beautiful piece of derived structure. Here is the Q of the GGQ I've been talking about, and he could see a density of a water molecule that was being held by the GGQ over the bond that was to be hydrolyzed at termination. This was between the tRNA and the completed polypeptide. So what that's showing is the chemistry of the vent in action structurally. Absolutely beautiful picture. So I'm going to now move back in time again, because we had projects going that were overlapping. This takes us back to the early 80s, when I got a chance to do our first and exciting cloning experiment, genetic engineering experiment, in collaboration with my postdoc lab. They had obtained the plasmid library called the John Carbon library of E. coli fragments. And so they attempted to kind of do a crude immunological assay to see if they could identify what clone might be release factor 2, and it was pretty messy. So they sent over us about 20 or 30 plasmids from clones that were kind of promising. And I'd employed a young technician, 18-year-old Tina Edgar, in the 1980s, and she had superb technical skills from the beginning. She retired this year after 44 years of working with me. Very sad day. So she grew up these cultures, and it was the fourth one she tried, made a protein extract, partial purification, had 30-fold activity. So we were ecstatic about this. But the trouble is, when the plasmid was sequenced, it didn't have a big enough open reading frame, that's between a start codon and a stop codon, to encode the release factor. Now, the obvious conclusion might have been that there was something there that was activating the endogenous gene, and that's why we were getting increased activity. I didn't want to believe that. So I did have some pure release factor, a few micrograms of pure release factor, and the ex-postdoc lab, Kesky lab, had just taken delivery of a protein gas phase protein sequencer, which was more sensitive. So I sent the protein over, and it was the first protein sequence on their sequencer, and they got enough. They got 44 amino acids. That was enough to tell us what was going on, and it was something quite amazing. And that is that we could match the sequence of the protein to sequence in the plasmid, and I'll just illustrate that here. So this was amino acid 21 leucine. Here's the code for it. Arginine, here's the code for it, and this was in the plasmid. 23, 24, 25. When you got to codon 26, there was a stop codon, and you got the next amino acid by slipping forward one nucleotide. Now, this has never been seen before. A stop codon had never been seen in a coding sequence before. And then you just went on and you made the protein. So this, we published this in PNAS, and it got a lot of interest. And so within a year, it made Watson's fourth edition of the molecular biology of the gene. So that sort of, I think, was an indication of the interest. So we spent a lot of time then working what was going on. When you looked at the coding sequence above, there were some interesting things. There was a sequence here which looks like what we call a Shine-Nagano sequence, which actually you find in head of initiation codons, which binds to the ribosome RNA and stabilizes it. So everybody who was interested in this around the world thought this was the key thing that was causing this event to occur. But Joanna Williams did a master's using eukaryotic ribosomes that don't respond to a Shinon-Dalgono sequence, and showed that frame shifting still occurred with good frequency. So we had good feeling that wasn't the major thing. And then there's in white there, I've got CUU, a leucine codon. Reading the literature, some people that played around with these sort of called the tRNA for that a shifty tRNA. So Stephen Robertson came and did a BMed Sci Honours with me in 1987. I gave him an impossible project. Somehow he still managed to get a distinction, and I had a few nightmares. I thought I'd ruined his future academic career. Fortunately not. But Cam Donnelly, a postdoc from Kennedy, worked on what was going on here. And let me just briefly tell you. So the ribosome was reading along the code and putting in the amino acid. When it got to the stop codon, it had two choices then on what we saw. Normally, you would expect it just to stop there and make a 26 amino acid peptide. In this case, so that's termination. Alternatively, it shifted and carried on making the protein, which was release factor 2. And this is the key. Release factor 2 is the decoding protein for UGA. It's the only protein which decodes tRNA. So this looked like an exquisite mechanism for this gene regulating its own, synthesis of its own protein. What Cam showed, that this was like a rheostat. We could modulate the levels of the factor, as might occur in the cell, and go from having 0 frame shifting to 100% frame shifting. So one wondered, is this a once-off in biology? Had we just serendipitously stumbled upon this? But of course, nothing's one-off in biology. And within a year or two, it was showed that viruses use frame shifting, but they slip backwards. They use it so they can make their structural proteins and their enzyme proteins in just the right ratio. And so the initial one found was RAS sarcoma virus. And so characterized here with a slippery site, a run of the same, more or less the same nucleotides. They had a stop codon like we found, and then they had a secondary element afterwards. But then when HIV was shown to have it, they had a glycine codon instead of the stop codon. We showed that in our lab that was absolutely necessary. And SARS-CoV-2 virus also frame shifts in the same sort of way. So we mounted quite an extensive program looking at the HIV. Quishla McKinney, the last part of her PhD, developed some enzymes, some assays where we could look at that. And then Jules Horsfield did a PhD on this. And of course, our bias was the slipping occurred at the stop codon, but the HIV people said it was simultaneous slipping mechanism on the banana skin there. So what Jules did with her PhD was just determine in great detail the slipping mechanism on HIV, and came up was that it was a what we call post-translocational. In other words, it was on the GGG, wasn't what was being believed everywhere. So that was quite controversial, but a publication of her, a main paper from a PhD in nucleic acid research, despite the controversy, was rated by the journal as being the top rated paper. We got a nice letter from the journal about it, and I thought I'd given it to her, and she thought I'd held on to it. We've currently lost it, but when I clean out my office in the next couple of days, I hope to find it. Caelan Crowe McAuliffe, when he stopped his PhD at Cambridge, came back and worked with me for a while. He looked at the peptides that were produced and showed and got data that supported Jules's mechanism from a PhD. So because AIDS was still in the news, and this was an area of HIV biology that hadn't been exploited, we initiated a drug screening program to see if we could find drugs that would inhibit this frame-shifting mechanism of HIV. Tony Cardno on the right there, he went over, we formed a collaboration with the WEHI drug screening section. He went over, embedded in there and worked for 2 years with their special library and isolated some lead compounds which would inhibit HIV. And I want to just go back. Since he started, a human gene had been discovered that used frame shifting. That was human gene PEG10. And Yosuke Shimaki did his PhD on that. But he was also able to show that the lead compounds didn't affect frame shifting in this gene as they had in HIV. So that was very promising. By the time we got to talking to biotech companies, AIDS had sort of just gone out of the news, rather like SARS has now, COVID-19 has now here. Biotech companies were talking about wanting to have drug programs which would eliminate the virus rather than just suppress it. And there was some change in leadership at the Walter and Eliza Hall, and they weren't quite so enthusiastic. So although there was a Japanese drug company which was interested and said, if we could convert our assays into a whiz-bang assay with fluorescent thing, they would be really interested. So go away and come back and do that and we'll talk. But I thought, well, they haven't offered us money to do that. And the climate was changing. So I decided to step back from this. And that was disappointing, but I don't regret having gone through that path way. When you get into this can be incredibly time consuming and energy sapping. And I still had things I wanted to do in terms of the other developing human disease work. So we stopped that. The last thing I wanted to section I wanted to talk about was on the recognition signal for the release factor. I said one of the questions was, did the release factor actually recognize more than the stop codon? And the catalyst for me to investigate this was Chris Brown, and between his master's and his PhD, he had by then become fascinated with sequences and signals. And although bioinformatics, I think, wasn't a term that was being used there, essentially he did the first bioinformatics. So we got the help of Peter Stockwell, who wrote some code for us, so Chris could pull out the sequences around the stop codon for particularly the E. coli genes, which were starting to be put into an E. coli sort of database. And so Chris analyzed those with this kind of analysis you see in the slide here. It wasn't the exact one, but the output was very similar. So it was looking at whether there was non-randomness on either side of the stop codon. In other words, if it was just the 3 nucleotides recognized, you might expect just a random thing, and then you come to the codon and it would be an Empire State Building, then it would go there, and then just random again. But you can see from this profile, as you got nearer the stop codon, the last 3 nucleotides, that's 1 codon, was somewhat non-random. And then you went on the other side of the stop codon, and you see what we called the 4th base, the base following, was very non-random. And then there was also non-randomness as it went down, actually up to 9 nucleotides from the start of the stop codon in the eukaryotic system. So fortunately, the frame shifting work I'd done had invoked enough interest that I got a Howard Hughes International research scholarship lasting 5 years and $1 million. And so I was able to employ Liz Poole, who had just done her PhD with Ian Lamont after working in clinical virology. So her task was to do the biology around this observation to see whether it actually had any effect in biology. And I'll just show I'm showing here one excerpt from her work where here we go, where what she did was to create an assay where the only difference in the sequence was at the 4th base. So she had sequence before, the same sequence after, for each of the stop codons, and then varied the 4th nucleotide. And you can see here with the result, if we look at the termination efficiency, UGAU was fantastic signal. UGAG wasn't bad. UGAA was pretty awful, and UGAC was a disaster. In other words, the biology actually reflected what we were seeing in this bioinformatics. And Kim McCaughan actually looked at the eukaryotic system, and she used an in vitro assay that I had used for my postdoc, where you add the eukaryotic release factor and the stop codon as a tetramer, and looked at how efficient it was, and she showed there was huge variation of efficiency. But now it was the A and G that were the most efficient, and C and U the least efficient. So the same phenomenon, but different examples in the prokaryote and the eukaryote. Interestingly enough, in my postdoc, when I was working with assay, we couldn't get it to work just with a single triplet codon. You had to have at least one more base to get the assay to work. So it was sitting there in front of my eyes all the time from postdoc that fourth base was somehow important, but it took this time to actually find it out. So interesting enough, again, I've got a lot to thank Venki Ramakrishnan, because he came up with a structure in a Nature paper in 2015 of the eukaryotic release factor and some messenger RNA with the stop codon. And what he showed in this was that when the release factor recognized the codon, there was a kink in the messenger RNA, and the fourth base was dragged in to the space of the decoding site normally occupied by 3 nucleotides. So it provided a beautiful structural affirmation for what we'd shown biologically. Okay. So, of course, I'd shown that there was non-randomness beyond that, and I'll just very briefly go through this. Louise Major started a PhD, and she loved it because she was doing designer stop signals. So we thought that the abundance of the sequences around the stop codon reflected their strength. And so her hypothesis that she was testing was that she'd take the most abundant and she'd call them strong sequence before, strong sequence of, strong, stop codon strong, and down the bottom weak, stop codon weak, and then strong in the front and weak in the back, weak in the front, strong in the back. And so she tested those out. And here signal failure is a big peak. And so the ones that she had predicted would be weak, the weakest, in fact, we thought the weakest, just was hardly a stop codon at all. It just read through by 60%. And then down the other end of that, we had a couple of strongs. We used 2 sequences, not sure which was the strongest, and they were very good signals. So essentially, we could design a signal according to whatever strength, a bacterial signal on the basis of this. Andrew Cridge did his PhD then on the rest of the sequence for a eukaryotic system, and this is a very complex thing, but this heat map, all I want you to note is this line down here. Red is incredibly poor, and we can read that off. This is for UGA up, 4th base C, 5th base G, 6th base C, and he did 7, 8, 9. When we published some of this, but we had a lot of sequence data without a model. What did it mean? I mean, we understood the 4th base now, that it was dragged into the site, but why so far further down? And then I read a structural paper about the channel which the messenger RNA is in when it comes into the ribosome. And the sentence said, from the first position in the decoding site, there are 9 nucleotides in the channel. And that, of course, clicked to the fact that we'd shown there was non-randoms in that 9. So then we published a paper finally in 2018 with the model being that when the message is going through the channel, it can form interactions with the ribosomal RNA and the protein at the entrance of the channel. And depending on what those interactions are, will influence the efficiency of the termination signal. What that means, of course, is a different story. Who knows? Now, the most poignant moment, I guess, that draws a line under all of this work was the Royal Society Award ceremony in 2010 in Christchurch. It was between the 2 earthquakes. So we made it into the Christchurch Town Hall complex. The second earthquake in February destroyed it, of course. And for me, this was a kind of a special moment for a number of reasons. This picture, I think, epitomizes it. On the left there is Stephen Robertson, my BMed Sci honours student of 1987. And he won, he's now, of course, Cure Kids Professor of Pediatric Genetics, and he won the Liley Medal at that ceremony. George Peterson, of course, in the middle, was a proud father. He had got the Rutherford Medal in 2003 as the only Otago person winning the Rutherford Medal. And this was the ceremony which I got the Rutherford Medal, following in George's footsteps as the second recipient. So, that's probably another photograph I'll give to my grandchild. So I want to finish just by one slide on what I call the Tate modern. So there's been 3 strands to this. One is on working on the molecular basis of memory and Alzheimer's disease, and a 5-group collaboration led by Cliff Abraham. And I mean, that's been fantastic to work with that. And in my lab, rather a new kid in the block, she only came in 2021. She's only worked for me for 23 years now. Katie Peppercorn spearheaded that. And the focus was on a neuroprotective protein, and she developed a way of producing that protein in cell culture and human kidney cells, and established a human brain protein resource, which we really did initially for the group, but she sent this protein all around the world now on request. So that's a very warm period of my research journey. And then from 2011, I've worked on the pathophysiology of ME/CFS in New Zealand patients. That was started by Erin Sweetman, who was with me for 8 years, and then Tina has put a lot of time into that, and then Alex Noble, Amber Hallowell, and Angus Mackay have contributed. And then since long COVID came in 2021, we've worked on long COVID and its relation to ME/CFS in New Zealand patients. And Katie, after getting her PhD at the end of her time working on Alzheimer's, switched to and has pioneered really the work on long COVID. So just want to acknowledge really all of the postgraduate students that have inspired me. I mean, that's what gives me creative ability, is just to talk to students and get something, energy off them really. Collaborators all around the globe and through New Zealand. I think this department, because it's been my home of creative endeavour for 57 years, and I admire and respect so many of my departmental colleagues over that time. And just in the Tate modern phase, we've been funded by Alzheimer's Disease Association, and also many private donations from individuals and families, and we have a sincere gratitude to all of the ME/CFS and long COVID study participants who've been very enthusiastic about participating in our studies. So thanks very much. I'll leave it at that.
Right. So I think what can be said, it's just an amazing journey through your scientific career in biochemistry, Warren. Fantastic. And still going. So Warren was made Emeritus Professor in 2000. And I guess we didn't really do this then, partly because there were funny things like bubbles and gathering limits and things going on in 2020, sorry, when you were Emeritus Professor, not 2000, 2020. So it's really fantastic to hear the story now. Warren has taught many of us in this room about the ribosome and translation. A few of you have had to hear it from me, which is not nearly so good as hearing it from Warren. But I think I've been inspired by Warren and also the fact that we still have lots more to learn. And so thank you so much.